Welcome, everybody. Hello, I am Ramona Palombo, and today we are going to talk about the single-cell RNA sequencing wet lab protocols in the first part. In the second part, Vincenzo will talk about the quality control. So in this first part, I have a presentation. So maybe I can share the screen and then we can start. Just a moment. Okay. So, yes, it is okay. Just a brief introduction of myself. I'm a biotechnologist, but I am also a molecular biologist. And during my research experience, I studied a lot the RNA molecules, starting from the gene expression, so the regulation of gene expression by epigenetic modification and so on, but also the alternative splicing events, so the alternative splicing processes, and also the translation of the RNA molecules into proteins. And today we will take a general overview on the technique that we can use to extract RNA, but also to have a good sample for the RNA sequencing. So just a brief introduction about what are the differences between DNA and RNA. I know that most of you or all of you are aware of what are the differences between these two molecules, but just a quick introduction of these. DNA is an helix, a double-strand helix, and so we have two strands that are one complementary to the other, and the sugar bonds, so the structure of the DNA, relies on the phosphate group and the sugar that we have in this part of the DNA, and we have also nitrogen bases that are different from the sugar and the nitrogen base that we can find in the RNA molecules. In fact, in the DNA molecules, we have the deoxyribonucleic, the deoxyribose. Instead, in RNA, we have the ribose. And also in the base composition, we can find the adenine, thymine, guanine, and cytosine in the DNA molecule. In the RNA, we can find not a T, but so not a thymine, but a uracil. And these are the main differences if we talk about the structure, but also in the three-dimensional structure of these two molecules, we can find differences. As we said, as I said before, the DNA is an helix, so it has two strands. Instead, RNA is a single-stranded molecules, and these molecules is not naked like this, like we can see in the image, but there are several RNA binding proteins that can protect protect and cover the RNA molecules, and it can also have different structures like three-dimensional structures that can also help the molecules to act like, for example, some help in the recognition of specific structure or specific portion of other species of RNAs. And this is very important because if we know that in the central dogma of the biology states that the information goes just in one way, so we have an information that is represented by the DNA molecule, then the transcription allows us to have an RNA that can act like a messenger. So this RNA is then translated to give us a protein. And so each information that is contained in the DNA is then translated, is then copied into the RNA molecules that is translated into a protein. So in a sequence of amino acid that can be also analyzed by the RNA. So differences in the RNA sequence could be transcribed also in a different protein, and so the alternative splicing events that can give different kind of RNA starting from the same locus, the same region of DNA, is an example of what I say. If the central dogma says that the information flows only in one direction, we know that there are also some particular RNA that don't encode for protein. There are examples in RNA that are similar to RNA messenger. For example, they could be polyadenylated also and 5' capped, but they don't encode for protein. It is the case of, for example, long non-coding RNAs. And so we have different kind of RNA species. We can have RNA messenger RNAs that encodes for proteins, and they are 5' cap. They have 5' cap, or they have also a polyadenylation as a tail of polyadenylated tail. Sorry, and this kind of RNA molecules are the most studied because they can give us the information of what kind of pathway, what kind of biological processes are activated in the cells that we are studying. Then we have also transfer RNA that have this particular kind of structure like a clover, and they are very important in the recognition of a specific portion of messenger RNA that is called also codon, that is represented by 3 nucleotides. And then it can facilitate the recognition of this particular portion, and in the ribosomal machinery can act like a transfer. Okay, like the name can transfer the right amino acid in the specific portion of the nascent protein. Then we have also ribosomal RNA that are part of ribosome, and the small nuclear or small nucleolar RNAs that are instead some small RNAs that can have different function in the nuclei. In fact, they can also be part of the structural part of nuclei, but they can also take part into splicing processes. Then we have microRNAs that can act as decoy for messenger RNA and regulate their translation. And the last two are the circular RNAs and long non-coding RNAs. They don't encode for proteins, and the circular RNA derived the biogenesis of this particular species of RNA is related to splicing events, and the function of these circular RNA are still studying. And the long non-coding RNA are very similar to the messenger RNA because they also polyadenylated, but the function of this long non-coding RNA are still to be explored in detail, but they can regulate gene expression. But why it is important to understand what kind of RNA species we have and what kind of RNA species we can have in a sample? Because if you take a look at the graph on this part of the slide, so we can see that I'm sorry, just a step behind. So if we have different kind of RNA species, we have also 3 different RNA polymerase that can synthesize these different types of RNA species. And in particular, polymerase 2 polymerase 2 is involved in the transcription of this particular kind of RNAs. Messenger RNAs, long non-coding RNAs, small nuclear RNAs, and microRNAs. They could be very similar, but also can have different features. And if we see the total amount of RNA that could be extracted by a growing mammalian cell, we can see that the most abundant species in the total amount of RNA are the ribosomal RNAs. And they are like the 80% of the total amount. So just the 5% of the total amount of RNA species are represented by the protein coding messenger RNAs. And a small portion of them could be also small nuclear RNAs and long non-coding RNAs. But according to the assembly that is not the last one, but one of the last, so this that I wrote on the slide, the human genes that we can find a very high number, so we can have 20,000 protein coding genes. So the number of regions on the DNA that could be translated and transcribed and then translated are very high. And so they represent about the 35% of all the possible transcript in a mammalian cell. So the ability of the wet lab protocols and procedures to enrich the samples with the RNA species that we are interested in is very important, could be very crucial. So just a little deeper into detail, we can see that ribosomal RNAs, for example, are very long. RNAs could be very long in taking an average size of 6-7 kilobases, and they are well represented in the transcriptome. But the RNAs that encode for proteins, so the pre-messenger RNAs, could be very long, but they are low in number. So if I want to analyze the differential expressed genes in a sample, I have to be aware that other kind of RNAs are in my samples, and to load to be able to decrease the background noise, I have to choose the right protocol, the right procedure to have an enrichment of my sample in specific species of RNAs. And if we know that in our cells and in all the cells of our body, we have the same information, so the DNA that is in all the cells are similar, if we can say also the same. Instead, the transcriptome of different kind of cells could be very different, and the transcription information can reflect also the specific function of a cell type, but also the cell state, and can give us some information about the response to particular treatment or disease condition and so on. For this reason, gene expression studies have become a standard procedure after the advent of high-throughput transcriptomics approach. And we can have microarray technique that is based on the hybridization of the molecules, but also next generation sequencing analysis that are more that are faster than the first, and they are very also useful to measure, to detect some unknown transcripts. Because if the first is based on the hybridization, the second is not. And since 2005, the next generation sequencing technology overshadowed the microarray thanks to these different advantages like high sensitivity for biomarkers discovery and unknown transcript identification. I'm sorry, maybe it's not so clear. I'm sorry. And sequence technology. So what kind of sequence technology we can have, we can exploit. And in the recent years, we know that the technologies continues to evolve, and so we have different kind of machines that are able to measure and to detect a very high numbers of molecules at the same time. And also the cost of sequencing is dropping, so we are living a beautiful moment for sequencing technologies. The first one that I mentioned before, there was microarrays to detect RNA species in our sample. And in this particular technique, we have that thousands of microscopic probes are bound to a solid surface that could be glass or silicon chip. It depends, or microscopic beads. It depends on the platform that we are using. And then we have a mixture of RNA extracted by our samples that are led to hybridized with probes. So when we have a fluorescence, so we can read the fluorescence in one specific spot, we can say that there is hybridation, and so we can detect the presence of a specific transcript. And this is the reason why I started this presentation with the description of DNA and RNA molecule, because when we talk about the DNA, but also of RNA, we can be sure that a single strand can pair with another one. So the microarray exploit these features of these two molecules. But the next generation sequencing is similar to this, but in this kind of sequencing technologies, we have what we called the sequencing by synthesis. And I'll show you why we can call it like this. So this particular way to method relies on the Sanger technology. And what is a Sanger method? With Sanger, that is one of the first technique for this kind of sequencing, use the template that could be our template, so our RNA samples, and we can use a mixture of nucleotides that are normal nucleotides together with fluorescent nucleotides that have been modified and the lack of a particular 3' oxidryl group. In this manner, the nucleotide that is incorporated in the elongation of the transcript is not working. So we can not add another nucleotide, and the transcription is stopped. So we have different kind of transcript in length, and starting from the first one, so the shorter one, until the longer, we can have the shortest and the longest. We can have we can analyze the incorporation of different nucleotides basing on the fluorescent signal that we can find. And so, as we can see in the bottom part of the figure, we can have this graph with different peaks with different colors, and each color represents a specific nucleotide. Instead, with the NGS techniques, we have a mixture of nucleotides that have fluorescent dye, and but in this case we can we can bind the template to a surface, and then the extension that is made by PCR, like in Sanger, obviously, we can have the incorporation of the nucleotides, the fluorescent nucleotide, and with a microscope, we can detect each nucleotide that is inserted in the elongation of the transcript. In this manner, we can have that with multiple steps of PCR, we have included the different kind of nucleotides that are detected one by one. And so we have different measurement in the same time. So with Sanger technology, we can have the sequencing of small number of templates. Instead, with NGS, we can sequence different number of templates at the same times and in a very efficient manner. And all the reads that we have, because always, not always, sorry, but often the DNA or the RNA that we are studying are fragmented. So we have different fragments of the RNA samples or DNA samples. And these fragments that are then extended by PCR are called reads. And the reads give us also another feature of the sequencing procedure that is called also coverage or depth of the specific procedure. Because if we have a different number of reads that align to the reference genome n times, we can see that we have the n depth of the procedure. So we have a defined number of reads that can be aligned to the specific region. Obviously, we have a genome reference, we need a reference, and the reference choice is very important, because when we analyze mammalian cells, but we don't know the specific organisms that we are studying, there could be some problem. So we have to start with specific information about our workflow procedures, and then we will see it later. But so the reference genome is very important to be updated. So we have to be sure to use the last version, the last annotated version of the reference genome transcriptome. Then we can also visualize SNPs, so single nucleotides polymorphisms that occurs in the half of the reads in a specific portion of the DNA or RNA molecule, but also we can identify some insertion or deletion that we can call indels. And upon sequencing analysis, we can also have this other information. So let's go to the central part of this discussion, that is the RNA sequencing workflow. The slide is very clear and it seems to be not so informative. In reality, I think that a good designing of the protocols and also of the experimental procedure for a specific experiment could be the very starting point, the golden starting point. So we can say that if we set all the right conditions and also we identify what are our objectives and the number of replicates that we need to have a sure answer to our questions, maybe it's the good thing. So as the first step, I choose the definition of the objectives of the study. So I have to start when I want to undertake RNA sequencing analysis is to analyze the different kind of conditions that I can use and what are the best condition that I have to use. For example, if I want to compare the genes that are expressed in a tumor mass respect to the normal control, but the normal tissue, it is represented by what? So I have to set up all this information or this condition before starting the experiment. In fact, we know that the cells that are inside the tumoral mass are very different in terms of viability, but also in biological processes that are activated in these cells. So if I take just the edge of the tumor and or maybe just a portion that is not so similar in the other part, so maybe I can create confusion also in the analysis of the data. And also the control condition, like I said before, we can imagine that we are studying a lung disease or a lung tumor, and so I decide to use a small portion, so a samples that I can take from the other lung to compare it with the tumoral tissue. But maybe, I don't know, there could be a problem in the other lung, or maybe the tissue that I can take, that I can collect, maybe is not very similar to the tissue that is interested by the tumoral growth. So there are simple questions that I have, but I have to answer to this question before to start with the RNA sequencing analysis. And also the model identification. So when we choose a cell line, we have to be aware that cell lines remains in culture a lot. And so we can also have some particular modification of the cell lines in terms of genetic patterns, but also in the expression of specific genes. Maybe they are resistant to some particular treatment or stress conditions, I don't know. And maybe it is something that we want to be aware of. And also the number of replicates, because I have a statistical measurement before the analysis of this, the experiment helped me to use the right number of replicates, the right numbers of cells, and also, most importantly, the right numbers of organisms, model organisms, like, for example, mice or rats, I don't know. And we have also to be aware on the sample preparation, because, as I said before, there are different kinds of RNA species that could be extracted from a cell, for example. And I can choose to enrich my samples with specific RNA species, for example, polyadenylated ones. And with single-cell RNA sequencing, we have this kind of enrichment also in the part of library preparation, because we use a poly-DT primers. But if I want to decrease the numbers of different RNA species in my samples, I can also use some particular columns that can have an also the ability to interact, to bind specific RNA molecules and elute the other one, or I can also choose different kind of extraction methods, for example, in solution with triazole, instead to filter them by kilobases, for example, etc. But also the timing. So when I extract RNA from a specific tissue or from cell culture, they could be I could have different yields in terms of quantity of RNA, but also in the quality of the RNA, extracted RNA, because the stability of RNA is not so long, and we have to choose also the right condition in the extraction, just for timing, but also in the buffer condition and also in the temperature. So for the last two part of this slide, I leave it to the second part of this webinar. Because we will talk also about the quality control and the library preparation, and also for data analysis we have different several presentations. So when I compare transcriptomics technologies, I can have different kind of technologies that I can exploit. And for bulk RNA sequence, I can say that it's like a smoothie. So we can combine different kind of cells, and so I can measure just the average of the expression of particular genes. For example, if I'm analyzing the differential expressed genes in a tumoral mass, tumor tissue, I can have some cells that can have different patterns of gene or expressed genes, and so I can detect just the average of them. So I can have a cell that doesn't express the gene X, but have an higher expression of the gene zeta, for example, and other cells that can express no gene zeta, but high numbers of copy for the gene X. So in summarizing these two conditions, I could have an average of them. And so in this case, I can say that maybe nothing changed for these two genes. Instead, for single-cell RNA sequencing, we can evaluate the expression of specific RNA species in different kind of cells. So I have the number of this particular gene per cell in my sample. And we will see how to make it. And with high throughput spatial transcriptomics, I can add some information to the single cell RNA sequencing that is related to the specific position of these cells in a specific structure. So I can say that, for example, the position of cells that are more sensitive to a treatment is on the edge of the tumoral mass, for example. So in this case, I can visualize, I can identify the single cells also not just for the gene expression profiling, but also for its position in the sample. And so what are the application of single-cell RNA sequencing? The goal of single-cell RNA sequencing, like we saw before, is to measure the distribution of transcripts across population of different kinds of cells. And just a little brief introduction about this application. Single cell was first reported in 2009 in a study profiling the transcriptome at single cell resolution. But in 2013, single cell sequencing was recognized as Method of the Year in Nature for its ability to analyze DNA and RNA in individual cells, revealing biological differences. But recently, single-cell technologies have progressed, and now we can add some measurements, other omics measurements, to have also different kind of information on the same experiment. And just to give you some examples, we can exploit the single cell RNA sequencing to evaluate transcriptional expression changes, in cells that differentiate. For example, I can follow the differentiation of a cell lineage and so characterize the different states of differentiation during time. Or I can evaluate the cell resistance mechanisms, for example, using different kind of cells that are responsive or not responsive to a specific treatment, and evaluate them during the time or at specific timing with different concentration of the drug, for example. Or I can identify novel cell types and states of cells, and reveal also the heterogeneity of the tumor microenvironment, because as we already know, in the tumor we can have different kind of subtypes of cells, and so they are not just the cells that are in the tumor. We can have also blood cells, immune system cells, and so on. And also trying to define what kind of immune cells can enter in the tumoral mass, can exert the action in the tumoral mass, gives me the information of what is happening in the tumoral mass. And I'm sorry. Yes. So okay. Sorry for the change. This is one of the several studies, publication that we can see online, and this group used the single cell RNA sequencing also in two different approach. One is with the 3' RNA sequencing, and the other is full-length RNA sequencing. These two sequencing methods highlighted the same results, but they are very different in the procedure, because in the first, they started with the isolation of epithelial cells from the whole trachea. And in the first, so in the 3' RNA sequencing, we have the analysis and so the sequencing of just the final part of transcript. Instead, in the full length, we have the sequencing of the entire transcript. And so we can apply this methodology just to a few number of cells, but we can increase the number of genes that we are studying. Instead, in the first kind of method, we can have high number of cells, but we can study lower number of genes per cell. And they were able also to identify a specific subpopulation that in the whole sample preparation are not so visible, as not so easy to isolate. That is this one that is circled, that are ionocytes. So they were able to identify these subtypes of cells in the whole tissue. And so what is the workflow of a single cell RNA sequencing? So we start from a tumor tissue or a single cell suspension. We can use also laser micro dissection technique to obtain the dissociation of the cells from the tissue or from each other. The important is to have a single cell that is alive and and viable, and the integrity of the single cell is very important. So then we release the cell to obtain RNA. We copy this RNA with to obtain a complementary DNA molecule that is amplified by DNA by PCR. And then we have the library construction, then the sequencing of this library, this library, and then the analysis of the data. This is just another slide to say the same, that take this the same message. And as we can see, this is from Azenta. It is a company that performed this kind of sequencing. So we have always the isolation of cells. The customer can also freeze cells and ship them to the company that can control the viability of cells to have an high quality of samples, and then the preparation of the single cell libraries. And we will discuss also about this part. So, but starting from the sample preparation, we have to be aware of some specific features of the samples, and we want to have an high quality samples. So the general protocols are expected to be compatible with many, but not all cell type and sample types are good for the general protocols, and so additional optimization may be required. But some key considerations are that we want a minimal cell debris, so a minimal presence of cell debris. So it's better to have the integrity, to be aware of the integrity of the cells, because a cells that is stressed or have lost its integrity can release DNA or RNA in the solution, and so I can have a high background noise. So I want low levels of cell aggregates, because I'm talking about single cell RNA sequencing, so I don't want that cells are aggregated and analyzed together. And I want also high cell viability, at least 70%, to have a good quality. And non-viable or dying cells, as I said before, increase the ambient RNA contamination. So it's very useful to use wide board tips to minimize cell stress, or I can also filter or exclude some cell debris or cell aggregates by the sides, and I have to be very I have to pay attention also, in the media and buffer composition, because sometimes some additional protein can eliminate the aggregation of the cells, to minimize the cell aggregation, but high concentration of ions like magnesium and also chloride can also affect the PCR part. So I know that it could be very boring, but I will not read all the protocol, but I want to give you just some information, some hints about the wet lab protocol. So we said before that the use of whiteboard pipette tips can help in minimize the cellular stress. But one other information is also the specific gravitational force that applied to the samples, because some cells are very sensitive to these. So be aware that each cell types has its own gravitational force to be applied, and specific timing and specific temperature. We can use also nuclei to extract RNA. We can see it later. And we can add also in the solution some additional BSA just to eliminate to reduce cell aggregation. But this is for cell suspension preparation. But what about the tissue? Because tissue is an aggregation of cells that are strictly bind to each other. So in the first thing to do is to manipulate the tissue, wash the tissue and manipulate it with forceps to cut it as I can. And then put it in a dissociator, so in a mechanical disruptor of this cell-cell adhesion. And also I can use some enzymatic approach to eliminate some interaction. And also in this case, I can add some BSA, or I have to evaluate at the final part of the protocol also the viability of the cell by also some specific stainings. And I go very fast on this. So after the obtaining of the cell suspension, I can choose also to freeze cell suspension and then thaw it in the following procedure when I want to analyze this, the sample. For freezing and thawing condition, there are several hints and several specifications. So, for example, the concentration of cells to minimize the viability loss, also the temperature in the and the timing of storage could be very important. And I can use also cryopreservation solution like 10% of dimethyl sulfoxide in media or serum to reduce the disruption of cells. When I want to use tissues that are frozen or fixed, I have to be aware that this particular kind of tissue maybe have been processed before. And so the structure, but also the feature of this tissue are different from a fresh tissue. So for fixed sample, we have to be we have to control also the storage, the temperature of storage and the timing of storage. And for frozen tissue, nuclei isolation is suggested because nuclei are more resistant to the freezing and thawing procedures, and so they can allow me to obtain a good RNA quality. But if I use just nuclei solution, nuclei RNA species, maybe I can lost some information about RNA that are translocated to the cytoplasm compartment, and so I can lost some information. So be aware of this. And for formalin fixed paraffin embedded tissue, we have some specific information. So for optimal assay results, we need a specific number of cells. And also for human tissue or mouse tissue, we need section with specific specific length, we can say, of the sections. But when I obtain the suspension of cells, I have to check about the cell viability and aggregation, because cells have to be viable for this technique and to be entire. So I can use staining procedure to visualize cells that are alive or dead, or calculate the percentage of cells that are dead in a sample preparation. And so I can decide to exclude that cells from the solution, or maybe isolate just what I want. For example, some cells that express a specific marker on the surface, on the membrane. And so there are different kinds of cleanup methods that I can exploit. Maybe the filtering is the first one. So it can allow me to eliminate large debris and also cell aggregates, but I can do it also by density gradient. Density gradients allows me also to obtain specific cell types. In fact, this is a technique that is also used with PBMCs or blood cells, because they can be divided in different layers after centrifugation. And washes can avoid large or small debris, and also slow centrifugation make the same. For the technique, for the cleanup methods, I prefer cell sorting because they can also remove dead cells, but in the same time I can choose a specific subpopulation in the tissue or in the mixture of cell mixture that I used. In fact, as I will show you an example of this, but with cell sorting, I can isolate specific subpopulation of cells. For the visualization of cells that are alive or dead, I can use also a combination of different staining. One example is acridine orange and propidium iodide, because acridine orange is cell permeable, so it can stain nuclei, and so it can generate a green fluorescence. Instead, with PI, so propidium iodide, I can analyze just the cells that are dead, because PI enters just in the compromised membrane, so of dying or dead necrotic cells. And it generates a red fluorescence. So in the figure in the slide, we can see that in the bright field, we can see a mixture of cell population that are green. Most of them are green, so that are stained with acridine orange, and instead with propidium iodide I can see the dead ones that are low in number, so it is a good sample. With fluorescence activator cell sorting, that is the FACS that I mentioned before, this technique analyzes the physical and chemical characteristics of cells in a fluid suspension. And so I can have the information for a single cell using a detector that gives me the information about the size of the cell, but also of its granularity. And this is for the cytofluorimetric part, but the fluorescence activated cell sorting is able to analyze the cell, as I said before, but then with the electrostatic deflection system, I can deviate the flow of these cells and collect them in different fractions. In this case, I can be interested, for example, in cells that are green or red because I used a fluorescent label inside the cells or on the surface of the cell, and so I want to analyze this particular subpopulation. Or maybe there are something that is expressed on the cell membrane, some receptor, for example, and so I can detect just the cell that expressed this receptor. It is also important in living and dead cells because dead cells express also the annexin, so I can detect the annexin on the surface of these cells, and I can eliminate these cells from the populations in the sample. And so I can enrich the sample with living cells. But using this kind of technique, I have to use also specific buffer to preserve cell viability, to take them on ice, keep on ice, and also in the dark prior to sorting. And after separation with FACS, I should have a specific number of cells because I can count them with FACS also. So the final part, so we are just near the end of this discussion. The cell partitioning allows me to obtain a droplet generation. So in this case, as we can see in the figure, in the image, we have a cell in suspension that is in the flow, in a perpendicular flow respect to the other, in which nanoparticle or beads are present. Then the second partitioning solution that is like oil for the second part can generate the droplet. So in the same droplet, I could have a cell and a bead. The specific goal is to obtain for a single droplet one cell and one microparticle, but it could happen also that some droplets could be empty or can contain just one cell or maybe two cells, why not, or maybe just one microparticle. In this case, we lost some information, but all the droplets generation is made in some particular, in specific condition, just to obtain that the majority is like 90% of generated gel beads in emulsion contain no cells. In this way, we can assume that we have for the droplets that contain the cells, just one cell. And so when the cell is inside the droplets, cell is lysed and then the RNA is released. The RNA that is released then has to be amplified through the generation of the library. But to obtain a library, we have to use the reverse transcription that is made by an enzyme, a specific enzyme that is the reverse transcriptase, that use a template of RNA and a primer of DT to obtain the first strand that is a copy, so a complementary copy of the first strand of RNA. Then for further amplification with PCR amplification, we have also the synthesis of the other strands, and so the amplification of the same region of the same transcript. So for the library generation, as I said before, we have one droplet and one cell and one beads inside the droplet. Upon cell lysis, I have that the beads that contain some branches, each branch contain the same cell barcode for the same bead, but different UMI. These UMI are unique molecular identifiers that quantify the original molecules of RNA. So each branch contact or can be aligned, can pair with a specific RNA messenger. And so we have a polyadenylation that can pair with the poly-DT primer that can act like a primer to the elongation for the elongation of the complementary strand. In this specific part, so I have the amplification of the signal, and so I have the library generation. And for the UMI consideration, for the UMI features, I let the stage to Vincenzo in the second part of the webinar, but just a brief introduction. So with UMI, I can then normalize the transcript that I have generated with the library, and so I can obtain the number that are present, the number of transcripts that are present on a specific cells in a specific cell. So I thank you for your attention, and my take-home message, not messages, but message, it could be that all start with a specific question: what kind of information I will expect? Because we saw that there are many different kinds of RNA species, and I want to analyze RNA messengers, so I want to extract just polyadenylated transcript, just a little things to say that with single cell analysis, we enrich the samples with polyadenylated RNAs. So, for example, I could have RNA, messenger RNA, but also take in mind these also long non-coding RNAs are polyadenylated, and so I can have also different kind of information. Thank you again, and I think that we can have a break of 5 or 10 minutes, and then we can start again.
Hello, good morning everyone. So my name is Vincenzo Bonici and I'm here for the second part of this webinar of the first International BioHackathon of the Bioinformatics Italian Society. So I will share my slides. And in this part of the webinar, I will talk about data preprocessing and quality control. So as have you seen, there are plenty of technologies that you can use for sequencing, for single-cell sequencing, and in particular for RNA sequencing. They started many years ago, 2009. Last technologies are a few years ago, so they have different techniques, but only recently we can use UMI to identify the molecules, the transcript molecules that are in the cell that we are sequencing. Okay, so of course, for every one of this technology, you should want to have a specific processing of your data, because every technology is generating something different. Every protocol is generating something different, and you should want to have some specific for your protocol. However, this is very difficult to have, and what we usually do is to normalize, let's say, the set of tools that you apply to your data to get the raw data coming from the sequencing and to obtain something that you can use for extracting biological meaningful from your experiments. So usually this is a kind of pipeline, computational pipeline that you can follow for analyze your single-cell RNA-seq data. So you have a step of isolation, then you sequence your data, and then you obtain raw data, raw single-cell RNA-seq data. This is very similar to what you do for bulk RNA or any other type of sequencing that you might want to apply. However, some of the steps from now to below are very specific of single cell because of the type of technology that you are involving in your experiment. For sure, because we are dealing with the sequencing, the first step that we should want to run is quality control of the raw data, of the reads. So we want to filter, as Ramona said in the previous part of the webinar, we want to get rid of all the reads that may have some technical problems because of the sequencing errors or whatever. Once we have checked the quality of the reads, then we can start to use them for our particular purpose. In general, what we do is mapping the read to a specific genome or annotation or transcriptome, and then we want to quantify the expression of each gene that should be in our analysis. After all this, we need to run a step of normalization because especially in this type of experiments that are not bulk RNA, bulk sequencing, we obtain something, and we will see this later, we obtain something that is very specific very relative to the amount of biological material that is inside of the experiment. And we need to normalize the data in order to be at the same, let's say, scale, and so we can use all of them in the same computational experiment. So after that, you, let me say, optionally run an imputation step, because you always have missing data, especially in single-cell RNA-seq, because since you have not a bulk RNA, so you are sequencing the RNA of a single cell, and probably you are losing something, okay? And often you want to impute what you have missed by the sequencing. However, many scientists do not like the imputation for specific purpose, so it's good if you take care and if you manage it with a very precise way, because you can also introduce some artifacts that are not in your data, in your biological data. So usually this is optional. Okay, this step is optional, and you may want to run or not run it. Of course, it's very helpful if you just want to visualize something or your data, and it helps you a lot in this type of maths. But for any other kind of analysis that we are going to do, especially for statistical analysis of your data, please be quiet with this type of steps. Then once you have obtained the normalized data, for sure, you can run several different type of analysis. You can identify subpopulation of your in your samples. You can run a differential expression analysis in order to identify genes that are differentially expressed between two or more conditions. Then you may also want to recognize the trajectory, the genetic trajectory and morphological trajectory of your cells that are in your experiments, and you want to infer it because it's always inferred. Okay. Then you may also want to reconstruct the biological network that is in your experiment, so on and so forth. Sorry. And if you are able to run a full-length transcript sequencing of your data, you can also run additional analysis such as alternative splicing, allelic expression, RNA edit, so on and so forth. Of course, every one of the steps that are present in this pipeline can be done with a series of tools. Every computational tool has a specific limitation, but also it comes to you with specific advantages that you may want to have in your analysis. So if you multiply the number of tools for every of these steps, the combination that you can obtain is very large. So usually what we do, we only use the most used tools and the most used combination that we know that they are working. However, we are not limited by this. For sure, for example, here, if we are talking about read mapping, three of the most important, most used tools for mapping this type of data are Top Hat, STAR, and ISAR2, but there are many others. Or, for example, if we want to perform expression quantification, we have cufflinks, sim, string tie, and so on and so forth. Okay, so usually you know the state of the art of the computational pipeline, and you always have to understand what these tools are doing, what are the limitations of these tools, and if you can go optionally or hopefully combine the outputs of two or three of many of these tools in order to obtain your desiderated data. So we can also summarize the phases of data preprocessing in single-cell RNA-seq in four, let's say, phases. One is the data acquisition. So once you have the raw reads, you align them to a reference. It can be a genome annotation, a transcriptome, or something else. Then you need to deduplicate your data because thanks to UMI, you can identify duplicates in your data and you can remove them from the analysis. And then you run the quantification step. But of course, after you have run the quantification step, you run the further step of quality control. Here we are not talking about the quality control of the raw reads, but here we are talking about the quality control of what we obtained by aligning and quantifying the genes. So the question here is, are we dealing with the right data? Is that what I expect from the experiment, or there are some errors, some condition that I didn't take into account, and what I'm obtaining is very different from what I expected. After this, I can normalize the data, and eventually I obtain this special type of matrix that is usually called count matrix. What is a count matrix? A count matrix informs us for every cell that we have sequenced, the expression of every genes that we are taking into account. Why I say that we are taking into account? Because we may want to get rid of some genes that are in our genome, and we may want to select what is called a whitelist for many reasons that I will explain later. And this is actually, I give you a spoiler for the next, for the biohackathon that we have in June. This is actually the data that you usually obtain when you start to analyze. So the start of the computational analysis that you will perform in June and in Naples during the biohack. You can also run imputation from this matrix, and after this step, we have cell assignment. For example, you can cluster your cells in order to understand in an unsupervised way if they form groups. You can classify the cell with different references, or you can trace the trajectory, the morphological trajectory. And eventually, as we said before, you can run differential expression, you can search for marker genes between different conditions, or you can search for expression patterns that are embedded. For sure, we can image as many phases as we want. For each phase, you have specific tools, so on and so forth. So actually, your what you have to do for the biohackathon in present is more related to the phase 3 and phase 4 eventually. And here I just show you which are the classical steps. I give you a very brief introduction about what you can do from raw reads to process them, to verify the quality of what you are obtaining in order to get this count matrix that will be the start of your work for the biohack. So since this is a very brief seminar, I cannot give you all the information that I would like to give you. So I invite you, if you are interested in this part of the pipelines of single-cell RNA-seq, that will not the goal of the biohackathon. But if you are interested in this part, you may follow many of the tutorials and online workshops that are available for free online. I particularly like the Canadian Bioinformatics Institute that has also a YouTube page in which you can find those and on workshop and tutorials regarding bioinformatics. And every year they do one of these workshops that is for specific for single-cell RNA. Okay, so I take inspiration from this workshop also for some of the material that are in this presentation. So if you have, you can go to the website, you can reach bioinformatics.cha to the YouTube channel, or you can also go to the GitHub pages, and there is one specific repository for single-cell RNA. So this is what Ramona explained to you in the previous part of the seminar. You start from the isolation, you have the RNA, cDNA, and then you amplify it, you have a library preparation, then you sequence what you obtained, and finally you have this count matrix that here you can also see as not gene by gene, but UMI by UMI matrix, because you are able to identify every single transcript molecules. However, we will count the expression of the genes and not of the UMI. Of course, you can introduce a lot of errors in these protocols, and you want to deal with them and get rid with them for obtaining the UMI count matrix. This is what Ramona just explained to you. So essentially, you're sequencing. This is what you are sequencing, but important for us is that part of what you are sequencing is given by the UMI, the cell barcode that is for identifying the cell, the specific cell, and sometimes you also have additional barcodes, for example, for beta-X barcodes, and for sure you have the PCR primer of the sequencing. Okay, So many of the recent technology, especially 10x and so forth, they have this type of sequencing structure. It's a paired and reads. You have two reads in the two directions. So usually read one, it's 100 base pair, and it contains the UMI, the cell barcode, the PCR primer, and all that is needed to identify the specific transcript in that specific cells in that specific sample that you loaded into your machine. And then there is a paired reads that is in the opposite direction that actually is again 100 base pair, almost, and it actually contains the sequence of the transcript that you are sequencing through your experiments. This should be, but it's indicative, it can be larger sometimes, it depends on the protocol and the technology, 300 in total, 300 base pairs, so you may have gaps between reads 2 and reads 1, so you don't know what is in the gap. Usually how we quantify gene expression, starting from the sequencing raw data, we want to obtain the UMI count matrix by a series of steps. Usually we map the reads to a transcriptome, we assign the reads to a cell, and we count the UMI for each cell. Okay. Now here we need to make some considerations of what we are doing and what can happen during these computational steps that we are going to perform. Okay. First of all, we should ask to ourselves which version of the genome am I taking into account for several reasons. Usually you use the most recent reference genome, but in special cases you may want to use something. Then you want to understand the annotation quality, because usually genomes are annotated. That gives you where the genes are, when the introns and exons are, so on and so forth. And you may have alternative annotations with respect to the reference shared one that commonly used. So you should take into account also the quality of the annotation, but also, let's say, the content of the annotation, because you may want, for your specific purpose, have a correct white list of annotations that you want to take into account for your experiments, because you may want to discard some particular genes or some particular isoforms, for example, or some particular elements of this annotation that you for sure know that you should have not been sequenced during the sequencing. Ramon showed you that you can sequence different type of RNAs. Actually, you can target different type of RNAs. It also depends on the specific cell cycle, for example, if you want to take into account the cell cycle, so on and so forth. So there is a problem when you try to map the reads to the specific annotations, to the specific transcriptome, because you may want to discard from this reference of the mapping what you do not expect to be read by your technology. Okay. So, and another question that I think is valid for any type of sequencing that you are performing, even if it's DNA sequencing, it's multi-mapping reads. So usually you have reads that maps in multiple parts of your annotation, of your genome. Usually we discard the multiple maps, but sometimes they are very useful, and sometimes they help us to understand, probably in this case, that one transcript was low coverage. We don't want to get the expression with transcript, but it's the primary mapping for our reads, for some of our reads. But for these reads, we also have the secondary mapping with almost the same quality in another part. We say it should be better. And of course, you should always be aware that you may always have sequencing errors, even in the UMIs that you are taking into account. So for what regard the map of the reads to the taxonomy, you should do all this consideration. And for single cell data analysis, There are three main user tools. One is STAR, Solo. STAR was for bulk RNA and DNA. Now there is Solo for single cell RNA. It's a new tool specific for single cell. And Cell Ranger, that is actually a suite of tools which contain also an aligner for single cell RNA-seq. So I also would like to suggest you to visit some of the repositories or resources online that provide you pipelines for analyzing this type of data. One of these repositories is NFCore, and it gives you a pipeline for single cell RNA-seq that is shown in this slide. So we start from the FASTQ file that are the reads, okay, with the quality of the reading. So they are paired with the quality of the reading, and then we pair it with a text file, CSV file that that inform us about the sample. That is the sample sheet. So inform us about what we did in the experiment. So we have a kind of raw data. We run FastQC as usual for any type of sequencing in order to filter and to visualize the quality of the reads, and then we can start with our analysis. As I said you before, if you have time and if you have the resources, I think it's better if you run multiple alternative pipelines, multiple alternative tools, and you merge the result of all of these tools. Here is what they propose to us. There are at least 4 pipelines that are specific for single cell RNA. One is performed by using the tools Salmon and Armin. So there is always a reference index that is generated, then the count matrix is generated, and then there is a quality control. In this case, specific for this pipeline, but you can also use the Callisto pipeline, the Star Solo pipeline, or the Cell Ranger pipeline. So in addition, Cell Rangers also provide something for multi-ohm analysis that here is highlighted in yellow. For example, as Ramona told before, we can run on the same experiment, we can extract information about not only for the transcriptome, not only the transcriptome information, but also any other kind of biological information, for example, chromatin information, so on and so forth, and you can combine it if the tool allows you to do it. Once you obtain this count matrix, it can be in several different formats, informatics formats. The most used ones are HD, H5. It's a very specific format, but many tools that from now to on analyze bioinformatics data, they allow you to input to them H5. Or, however, you can have a specific format for the Seurat suite that you will see in the next webinar, or any other kind of type of formats. You can run a multi-QC at this time. Quality of your data is normal multi-QC analysis that you can find in any other tutorial, and you can make an idea about the quality here of your data. And of course, the aim of this pipeline is to combine all of the results into a single matrix that you can use. And in the specific of this pipeline provided by NFCore, there is one of the tool of the Cell Ranger pipeline suite that is called Semblender, that you can use in order to remove the background from your data. But this is just an example that I invite you to visualize. NFCore at the end is an open source analysis. They provide you a repository for open source analysis pipeline by built on top of this framework that is called Nextflow, and it's specific for describing pipelines. So you can have a look at the website. There are many pipelines that are available, and you can use them to have an idea of what is the current state of the art for analyzing a specific type of bioinformatic data, for example. So going back to our analysis, this is the type of reads that we have. Important for us is the fact that usually it comes in 3 files, the sequencing. You have one file, one compressed file in FASTQ format for reads 1, one compressed file in FASTQ format for reads 2, and a third file that is the sample index file. So this is very, very important. It's a third file that you want to use for dividing the reads by the sample that you have put in your machine, because you can run multiple samples at the same time and you need to divide the data. Once you have divided, then what you do within each sample, you want to group the reads by the barcode, the cell barcode. Once you group them, you want to align them to the transcriptome, or you can also sometimes run an alignment-free cell calling in order to understand what type of cells with what you are dealing with. And after you have run this step, you finally want to quantify the expression of the genes that are present in your cell. And this is actually what the pipeline of Cell Ranger is the today most used pipeline for single-cell RNA data analysis does. So in the end, what we have within each sample a set of reads. These reads contains the barcodes, cell barcodes, the UMI, and the real cDNA data. So first you group them, so by barcode, so you can distinguish them by the cells. Now that you have grouped by the cells, you can align them to the specific transcriptome that you may want to use. So for each one of these molecules, you know to which one of the genes in your transcriptome maps. Always remember that what you are aligning is the cDNA, not the wall information that is in the barcode in the UMI. Of course, you have to discard this part of the read, and you only map the reads number to your data. Then, after the alignment, you want to delete some duplicates, and you may do now it thanks to the UMI techniques that allow us to remove these duplicates and the duplicates the data in order to obtain a good realistic quantification of our genes. So be always aware that there are a lot of errors that can be introduced in this, and also errors that can be in your data, and they are intrinsic, like sequencing errors. This can discard some of the low quality reads, but some of the reads will always have some errors. And what is a common error is to consider the whole transcriptome, for example, of the human genome. Okay, this is not good, because you should have a wide list of the genes that should be in your analysis, and that you are going to sequence with your experiment. Here there is a very common, very famous example that regards pseudogenes. Pseudogenes are often included in the annotation of the genome. However, as you may know, they are not expressed because they look like genes, but they have no primers for the transcriptions. So at the end, if you consider them, and if you left them in the annotation that you have taken into account, to which you are going to map, some of the reads will map to the pseudogene annotation, and the fact that some of these reads will map to this pseudogene annotation, you will lower the abundance of the reads that are going to map to real genes that you are sequencing with your data. So this is just an artifact. Usually, if you put them almost 4%, it's better if you remove them from your transcript. So for what concern the reads and what you should obtain from your data, this is a receipt from Luciano Cascione, colleague of mine, and is head of bioinformatics at the core unit Institute of Oncology Research in Bellinzona. And the recipe is that the estimated number of cells that you are sequencing on average, depending on the protocol, of course, and on what you are sequencing, is between 500 and 10,000 cells. The number of the mean reads per cell should be 20K, so 20,000 reads per cells, but the minimum recommended one is 10,000 per cell. So you should have 10,000 reads mapping in each cell. It means for each barcode, cell barcode, you should have this amount of reads, but we will see this later. The valid barcodes should be greater than 75% because you may want to discard some of the reads because of the FastQC tells you that the quality is very low. But if the sequencing has performed with some problems, you probably many of these barcodes will be discarded by the quality control. If the percentage is below 75%, it's better if you think about what you obtained from the sequencing, if it's very good or if you need to rerun the sequencing. Q30 bases in RNA reads should be ideally greater than 65%. So the quality of your data should be very high, or the quality of your reads should be very high. And let's say that reads should be mapped confidently to the transcriptome, and the percentage of these that are very confidently mapped to the transcriptome should be at least 30%. So very high mapping, high quality mapping to the transcriptome. And usually, since you are sequencing the cDNA and not the reverse, the antisense DNA, the percentage of reads that will map the antisense of each gene should be less than 10%. So if you are performing this percentage, this calculation is near the percentages that are shown in these slides, you may have a good read, let's say, set of your data, and you may want to proceed with all the other step of your analysis that we saw before. So, but talking about single cell, what are the main problems actually of single cell is that last technologies, we have the oil, we have the droplets, so on and so forth. Ramona told you about this. Ideally, for each droplet, we have one cell, one BITS, that is a barcode, and a little bit of ambient RNA, because there is always a little bit of ambient RNA. And this is the reason why these patches, so this is one of the reasons why these patches are not 100%. But usually what you obtain is a large number of empty droplets, but so you didn't capture any of these cells, but actually you captured some of ambient RNA. Okay, and you will get some reads from your data, but these reads are not of your cells, but it's just from something else. Then in some cases you have doublets, which means that within the same droplet you have two different cells, but the barcode, the cell barcode, the bits is identical, so you cannot distinguish between them. And as you may want, this is a kind of bulk sequencing, but with just two cells, and it is a problem for quantify the gene expression. But at the opposite direction, you may have two barcodes, bits in the same droplet with just one cell. So what you obtain is that you split the reads into two different barcodes as it was two different cells, and you just half the quantity, the expression of the genes that you are extracting. And of course, you have some droplets in which you do not have the beads, so they may be just empty, or they may be with some cells. The problem is that in some situation, you get some reads from this, but the problem is that the read do not contain the barcode. It contains something else, and usually it's not a valid barcode if you're lucky. Okay, so usually you have the list of your barcodes or a way to obtain the list of your barcodes. It's not in the list, and you know that you can discard it. But if you're lucky, and if the probability to obtain a valid barcode is very low. So take into account that the doublet rates however, depends on the loading that you do in your experiment. So there is a proportionality between the number of cells that you load into the machine with the number of cells that are actually recovered, of course, but there is also a direct proportionality with the doublet rates that you are going to obtain. It's not just 0 point something, but sometimes it can be a very high percentage, because 5%, in my opinion, is very high sometimes. So, and if you see, if you are loading 8,000 cells, you can obtain almost 5% of doublet rates, and then you may have some problems in quantifying the gene expression. So usually, if you take the barcodes, so actually the cells of your experiments, after grouping the reads by these barcodes and by the UMI, and after the deduplication step, and you sort these cells, okay, according to the number of UMIs that are mapped to that cells, okay, you obtain a chart like this. So for the most common cells in the human body, this is the number of UMI that you obtain is between 10K and more, okay, for each cells, and this is considered normal cell. But of course, there are empty cells, so the number of UMI that you are mapping to that cells, that you are considering as part of the cell, when it's lower than 100 or 200, let's say, you may want to discard that cell because they were just empty droplets, okay? Of course, there are some of them that are with ambient DNA, but because it's ambient DNA, the amount of this ambient DNA should not be that much, so it should not be comparable to normal cells in the human body. It's just for the human body. But of course, not all the cells are the same. Some of them have low level expression of the RSAs, but by themselves. And for sure, there's part of this chart in which you can identify, let's say, low RNA cells. Okay. So what actually the tools are doing is they are going to filter to get rid of some of the cells, namely some of the barcodes, by the UMI count. Okay? This just there is a new version of Cell Ranger, the 3, that now takes into account low RNA cells. Before they were discarded, and the pipeline was taking into account only cells with a normal, normal amount of RNA. So how we could now measure actually the quality of what we obtain, and after these steps of grouping, UMI, filtering, so on and so forth. We actually, there is not a specific thing that we can do. It depends on the experiment, the experimental setup. It depends on what we wanted to sequence. It depends on what we put in the sequencer, so on and so forth. But of course, here there are some indication of what we could take into account for evaluating the quality of the UMI count matrix. You can take into account something that is related to gene expression. Are there some apoptosis-associated genes? If we, for example, we are planning an experiment regarding cancer or this type of disease in which apoptosis is essential. Are there some housekeeping genes? If we know that the type of cells that we are sequencing as an already known specific housekeeping genes or this kind of things, you may just check that these genes have not zero expression, but they have a valid amount of expression. You can also compare cytoplastic between membrane genes. You can see the level of ribosomal genes, mitochondrial genes, metabolism, genes related to metabolism, and so on and so forth. Okay, so you may also want to, of course, look at the percentage of reads that are mapped, and you can also want to understand the level of high variable genes that are in your data, lowly expressed genes, genes that have been deleted, so on and so forth. So this is usually something that you do by experience, but you may have some indication if you want. Okay. So actually, it turns out that the good method that is always valid for understanding the quality of your data is to measure the mitochondrial DNA genes, and depending of the expression, the expression level of this specific type of genes, you may want to you can understand if you have an high quality sequencing, an high quality UMI counting matrix. If you have multiple drop doublets, or if you have a broken cells, or if you have an empty cells. And actually, as you can see in the paper, you can have a valid indication of it. Okay. So be also aware that you have a problem linked to the library size. We usually assume that each molecule has equal chance of being sequenced. However, it's not true, and it's almost proportional to the number of reads of your experiments, the total amount of UMI that you obtain, but it's not always true. So you may want to have different setups depending on the sequencing depth that you use in your experiment. And usually what we do to normalize this data, we simply divide each cells by the total UMIs count that are in your cells, and you obtain a kind of percentage of the total RNA. And then since these numbers are very low, you multiply these numbers for example, 10,000 in order to obtain normal looking numbers. However, you still have a problem of proportionality if you have genes that were considered very expressive with respect to the other genes. So you may want specific tool that may deal with this problem of proportionality. So here is one of the difference between bulk RNA and single-cell RNA. Usually in bulk RNA, an assumption is that most of the genes are not strongly differentially expressed between cells. However, for single-cell, this is not probably the case. So tools such as DSEC or TrimMed are not suitable for this, for analyzing single-cell data, because they do this assumption. So you may want to consider the specific tool. In this case, for example, we can suggest SCRAN for removing this problem of proportionality. Okay. You may also want to take into account the fact that the behavior of the genes, so the expression of the genes, it's not always the same depending on how the total number of cell UNI that you have in your experiment, so the number of cells that you loaded, so on and so forth. So some genes, they look like the gene expression is proportional to the total UMI count. Actually, if you log normalize them, you see that there are some very difference. And usually the tools have to take into account these facts. One of the most one of working technique is to use Pearson residuals. It's a very specific statistical way for assessing, for normalizing, transforming your data such that at the end of this transformation, your data should have a shape that is the shape that is expected by the statistical analysis that you are performing, because statistical analysis, they always have some assumptions, such as the shape of the distribution that you are taking into account. It should be a normal distribution, but especially for single-cell RNA data analysis, for many reasons, this at the beginning, the raw UMI accounts are not normally distributed, and you need a way to, let's say, make them normally distributed, and you can find some additional information about this in the tutorial. So at the end, we through these steps we obtained these normalized expressions. So we have cells and genes. For each cells, we know the expression, the level of the expression level of each gene. It can be the opposite. You have cells as rows and genes as columns. And from now on, you want to perform your analysis, your real computational analysis in order to obtain something that has some biological meaning. Some of these steps can be feature selection. You may want to select some of the genes that are of interest for you, but most important, you may want to run dimensionality reduction, or you may want to calculate cell distances. And once you have these information, so the similarity or the distance within all the cells that are in your experiments, then you are able to do differential expression, clustering, trajectory, so on and so forth. And all of this will be part of the next webinar that will be presented online for the BioHackathon, and they will be part also of the BioHackathon. So this is, I also suggest you another online resource that is singlecellcourse.org, in which you can find many other consideration and indication that you can take into account for this step of the analysis, that is from the raw sequencing data to the count matrix that you will use for your analysis. So from my side, it's all. I thank all of you for attending this webinar. I also would like to thank all the organizers of the BioHackathon, and also all the organizers and members of BITS that will allow us to do this BioHackathon for BITS in this June. And especially want to thank Ramona Palombo for being with us today and for being part of this webinar. And I also would like to thank Salvatore Calderaro, that without his support, we will not be probably able to stream this webinar online and register them in order to be available to you. So thank you very much.